So I will share with you the screen that we left last time in the class, and that was gRNA designing for plants. So that's in fact the gRNA designing. We will be considering both the plants and animals. The first is actually about the gRNA designing for plants. That is part 3. This is one of the examples we are doing at FCC, okay? So what I am showing you in this slide, this is the gRNA designing for plants, okay? And what is our motive? Our motive is that this is the typical wheat which is grown in Pakistan, okay? This one, this is hexaploid wheat, okay? And this is tetraploid wheat, which has a mutation in a gene known as BHT gene, okay? The mutation is that the nucleotide T at position 287 in the gene is mutated to C, adenine to cytosine. So it is changing the amino acid at that position, amino acid number 96, from leucine to proline. A single amino acid change is resulting into this format of wheat. Okay, this panicle of wheat has so many branches. That is the branch pattern. And this panicle contains much more number of wheat seeds as compared to this one. Okay. So if we want a revolution in the production of wheat, then we should move from here to here, because the system naturally exists due to this mutation in the tetraploid wheat, which is used mostly for making the pizza dough. Pizza dough is not from hexaploid wheat. Remember that. A good pizza dough is always from tetraploid wheat. Okay. So if we make this mutation in the hexaploid wheat, there are chances that we can get a hexaploid wheat with the same branching pattern. Okay. So single mutation is equivalent to repairing the gene at a very specific point. That can be done by CRISPR-Cas technology. Okay. So for that purpose, what we need the nucleotide sequence of the BHT gene. This is the BHT is a branching factor protein. Okay. It's a branching factor protein which induces branching at the nodes. Okay. And this is what I was mentioning. This is nucleotide number 287. In hexaploid wheat, it is T there at 287 position. In tetraploid wheat, it is C there. Okay. And as I mentioned earlier, it is a mutation from leucine to proline due to change of the single amino acid. All the rest of the nucleotide sequence is the same. So is the protein sequence. One mutated amino acid, one chained amino acid causes this protein to cause these branches. Okay. Okay. So now I will ask the students that if you are required to change this nucleotide from T to C, and you have gone through the CRISPR-Cas lecture. You have gone through non-homologous end joining and homology-based recombination. Okay. What would be your approach? And you know that you can design a gRNA. You can select a protospacer for the gRNA from the nucleotide sequence of the target gene. In this case, this is your target. You want to mutate here. What will you do? You will select the protospacer, definitely. Suppose it is SP-Cas9. SP-Cas9 has a PAM site which is NGG. Okay, and you will paste the sequence on the Word file, and on the Find menu, you will search GG, and then you will mark every GG on the sequence close to the position where you want to do mutation. Okay, so are you in green? Whatever you see in green, it's a PAM site. GGC, GGC, GGA, GGA. So these are all PAM sites on the sequence which are highlighted. Not all actually, you can see GGC here as well. GGC. So I have just randomly highlighted them in green so that you can visualize that SP-Cas has so many PAM sites on our target sequence, close to our target as well. Okay, but what would you do with all these PAM sites? You have identified the PAM sites. So the next thing that you will do is, क्या करेंगे आप? What will be the first step? First step, you will remember the 4 rules. If you remember the 4 rules, that the nucleotide closest to the PAM site should be either an A or G, right? सो गए या चले गए आप लोग?
Yes, sir.
सिर्फ आप ही हैं लाइन पे? There are 20 students, and at the moment there are 16 online. The closest. Okay, so answer me that. The first rule of selection for a protospacer is a 20 nucleotide sequence that you select on your target sequence for making the gRNA. You select the protospacer close to the PAM site after identifying that where are the PAM sites located. The first nucleotide upstream to the PAM site should be an A or G. This is the first rule.
Sir, A?
A or G, which depends on the type of promoter that you will be using on the vector, U3 or U6. U3, you will have an A. For U6 promoter, you will select a G. Is this protospacer on this PAM sequence okay or not? It has got a T here.
No, sir.
Okay, it's not a good protospacer, right? What about this one? There's a C here. Again?
No, sir.
What about this GGA here? There's an A here.
Yes, sir.
Yes,
It's a
sir.
good
It's a
one.
good.
If you are
Yes,
using,
sir. It's a good.
okay, that's fine. That's excellent. If you have a U3 promoter on the vector, then this is a perfect place to select them. So what you will do? You will count from, no, it is not. This is NGG is actually the PAM site. The next one is C. It's not. It's not. Again here, GGC is actually NGG. What about here? NGG, again C. And you need to look for NGG, again a C here. NGG, a G here. Yes, this one. This one. This is NGG. And on the top of this N is the G. And this is good for U6 promoter. Is it clear? So you
Yes,
got,
sir.
okay, let me escape from here. I hope you can still see it. And if I can take a drawing tool from here, go to chup care, is behind that. Okay, let me try. ये क्या करता है? Hide floating meeting controls. Okay, so if I take this one and if I mark here, if I have this NGG selected here, this one, okay? So you can see that this NGG, on the top of this NGG is a G, right? This one.
Yes, sir.
This is a G here. It means that this sequence is perfect for a U6 promoter. Okay? So what you will do, this will be your first nucleotide, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, and 20. Okay. So the first thing that you are going to do is, this is your protospacer. Okay, shape fill with yellow. Oh no, selected everything. So these 20 nucleotides, they are actually your protospacer sequence. It's not actually fill. I should color these in blue, dark blue, or green color. This brown. Oh, purple. It's not that clearly visible. This one is okay. Okay. Now, if you have these 20 nucleotides on the top of NGG, this is the PAM site, and the PAM site is always on the target sequence. It's not the part of gRNA. Okay? So what you will do? You will take these 20 nucleotides. Look at position 17 or 16. Okay? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, and is 17. Yes, T is okay. It should be an A here in case of U3 and a T in case of U6. Okay, so we have a T here. It means that this protospacer is really going to be great. Okay, we have a G here at position number 1, and we have a T at, and we can use U6 promoter. I mean, we should select a vector which has got U6 promoter for the expression of this gRNA. Okay. So the first thing that you do is making the protospacer. Okay. Deciding a protospacer. How many protospacers you will design on this sequence? And what would be our strategy? We will design at least eight protospacers upstream to this region, because one of the gRNA will make a cut on one of the protospacer, right? If it is CRISPR-Cas9 and the Cas9 is SP-Cas9, it will make a cut. Where would it make a cut? Bole. At this position, 3 nucleotides away from the PAM site of target. Do you remember that? Okay, here will be your one cut. And if we are using natural Cas9, it will make a double-stranded cut. If we are using a CPF1 enzyme, it will not recognize this PAM1. It will recognize VTTT. VTTT. Okay, that's another PAM sequence for CPF1. So if you are using a CPF1, then you don't have to look for NGG sequence. You have to look for a triple T. And on the top of the triple T should be A, C, or G, not N. Okay. So if you study that slide again, you will see what is VTTT, V triple T. Okay. So I'm taking the example of SP-Cas9, which is natural, which will produce double-stranded DNA cut. This is single-stranded DNA sequence. If you make a double-stranded, the other cut will be just opposite to this one, opposite to this arrow on the other strand. So you will get a blunt-ended cut on this side of the sequence, which is here. We want a mutation here. So you will make a cut on one side of the mutation, and then you will also design another protospacer on the other side of this mutation, which is below that sequence. What about this one? What about this one? So this is NGG and then G. You see, there's a G here. U6 promoter. U6 promoter. Another G here. So we count 20 nucleotides here. This is 1, 2, 3, 4, 5, 6, 7, 8, 9. Oops. 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. So from here to here, there will be an other protospacer. So one protospacer here, it will go to one gRNA. This protospacer here will make another gRNA. So that the protospacer, CRISPR RNA, and tracer RNA complex will bind here along with the Cas9 and will make a blunt-ended cut here. Similarly, the second gRNA will bind at this place and will make another cut. Where? At this position. Okay, here. So you What will happen when you make a cut on the DNA at this position and this position, and the cut is double-stranded? This small piece of DNA fragment will be lost by that chromosome. And if we introduce a new DNA fragment at the moment of cutting this by the Cas9 with the mutated T, we replace that with C at position 287 on that fragment. There are chances that that fragment of DNA can come and can ligate at this position, thus mutating the gene. Do you pick my point? You have to delete the fragment which is having the point where you want to do the mute modification. On both sides, you cut that. So you decide which CRISPR-Cas system or CRISPR CPF system you have to pick, blunt-ended one or the one with staggered ends. Okay, so I would prefer to go for staggered ends, the modified versions of CRISPR having dCas9 along with fork 1 or Cas9 having mutation to cut on one strand, and then we have to decide another one, but that cannot be applied here. So it's better to go for CPF1 to make a 4 nucleotide or 5 nucleotide overhang on this end, overhang on this end, and then you have an overhang on the fragment where you have done the mutation and you incorporate that in the cell so that the repair system, the homology-based repair system should get active and that fragment can be inserted at this point. Are you clear or not? If you have.
Yes, sir. Clear.
Yes, sir.
Okay, that's perfect. So now if somebody asks you that how would you start your experiment on CRISPR-Cas9? So what would be your answer? Your answer should be, I will get the target sequence first. I will identify where to make a mutation. Okay, so around the mutation, you will make the protospacers and you will add those protospacer sequences to the gRNA. You will make 2 different gRNA, one on one side and other one on other side, okay? So that you can replace, you can cut off that fragment from the genomic DNA, and then you will insert an identical DNA fragment with the only difference of 1 nucleotide change or 2 nucleotide, whatever you want to change, so that the genomic DNA can get that fragment. Obviously, when you cut one genome, there will be one fragment out from here to here. And if you want to replace it with another fragment, you introduce millions of those fragments, so that there should be a chance that from millions of the fragments, only one can get inserted there. Okay, this is a great efficiency, but this is just at one point. The drawback of genome editing is off-target sites. Okay, CRISPR-Cas has much less number of off-target sites as compared to the zinc finger or talons. Okay, so this is much more efficient, and the efficiency improves when you select the right CRISPR-Cas system. I should not say CRISPR-Cas system. It may be CRISPR-CPF1 system and their modifications. So you need to study the literature and look into the different versions or modifications of the systems of CRISPR, okay, and select the best one which will do the job most efficiently. That's what is the science. That's what is a graduate student, a technician can do. Just they can pick a Cas9 and do that, and then there will be a failure. The one who is educated for this, he will or she will look around all the best systems and decide which one to use for introducing that mutation very accurately. And the same system can be introduced to put actually, instead of putting the fragment with a single mutation, you can insert there a whole cassette. That's equivalent to developing a GMO. Okay, so can you make a cut? You introduce the same nucleotide sequence that you have removed, except with a mutation, or you can introduce a whole cassette downstream to a promoter, or you can have your own promoter in the cassette, and you can put it in a place where no other gene is affected. And this is how the CRISPR-Cas system works. Okay. Now this was the theory, and I hope that if you have to begin, you can think that from which point to start and where to end. Okay. Now, at this moment, I realize you have a question in your mind that, okay, I will select this protospacer. Then I'm going to make a gRNA molecule. Okay, I will tell you shortly. So don't worry about that. But how you can use the available software to do the same thing that you are doing manually. Okay, knowing or having the knowledge by doing the things manually is always very important. It makes you perfect. If you are using just a software, you will not know what the software is doing. Okay, but you know that you are selecting a protospacer, you are making a consideration that the first nucleotide close to the PAM site should be a G or an A. Okay, at position number 17, it should be a T or an A, depending on the promoter you are using. And the same thing will be done by the software as well. So software will do immediately, and then you will look into the protospacer that the software has generated. Does that PAM site has the same A or G at the first position or not? This is what you are going to analyze afterwards, after the software proposes you with the sequence of the protospacer. So let's see how the things work. If you know about the Ensembl plants, this is what we are going to do for designing a gRNA for plant system. Okay, you will see how simple it is. Okay, it's very simple. You go to Ensembl plants. Okay, then if you click here at this position on this arrow, where you see all species is written there, you will open up this link or linked window. Okay, so if you scroll down, you will see the names of every plant that has been, whose genome has been completely sequenced. So what it represents is a whole genome of Arabidopsis thaliana here, okay, Brassica napus here, Brassica repa here. So you select the plant on which you want to do the genome editing experiment. Okay, Triticum aestium is here, and this is wheat. So we want to do that mutation in wheat, okay, in the BHT gene. So what we select is, we select Triticum aestium. The first thing after opening this, here you see Rhiza stiva, the rice is here as well. Hordeum vulgare is also here, okay. So you can see the Triticum directly on the main screen. Gene is also there. the model plant reviewers. So you can select from this menu, or you can, if your plant is there on the top screen, you can just click on there and you enter into that plant, Triticum estimum, and you click on go, and it will go to the genome. So what you do now, this is, for example, one of the protospacer that you have designed. It's not a 1. You can see a greater than sign on the top. Okay, it means that you have selected 8 or 10 protospacers. So you can put those on a Word file, okay, in FASTA format. This is known as FASTA format. A greater than sign followed by the nucleotide sequence. Again, you enter, put a greater than sign, and then you enter another sequence. And then you enter and put another greater than sign, and you put the other protospacer sequence. So you can simultaneously paste here 10 or 20 different protospacer sequences separated by a greater than sign. Okay, all the protospaces you have already selected Triticum estimum. Okay, and all those other things are the default values. You just click on run. Okay, so when you run, what you will get there is that you will get the output. Okay, so this is actually protospacer sequence 1 that you selected. Then this is protospacer sequence 2, 3, 4, 5, 6, and 7. Okay, so what I mean, actually, there were 7 protospacer sequences which were pasted here, separated by the greater than sign. Okay, so you get 7 outputs. Output number 1, what it is telling you, let me enlarge it. It says 11 hits found on the Triticum estimum genome. Okay, 11 hits. For the second protospacer that you selected, the blast search, that what you had done actually by clicking on the run, you initiated a blast search on Triticum estimum genome for the presence of those protospacer sequences. The first one is found 11 times on the genome of Triticum estimum. The second protospacer that you made, it matches at 37 different positions. Actually, what should have appeared there? Now, wheat is hexaploid. It means it has 6 genomes in it. Actually, 3 genomes and the duplicate of that. So how many similarities should you have found in a hexaploid wheat? 6. Okay, the maximum number here should have been 6. Okay, why it is more than 6? There are 5 more than 6. Those are the non-specific sites. Means your protospacer has identical sequences at 11 places, but those about 5 of them, they exist in non-BHT genes. That's the reason it's called non-specific sites. Off targets, these are called off targets, that if you mutate BHT, other 5 genes on the wheat genome will also get mutated. Okay, that is the problem. You cannot restrict your genome editing to a single gene. Your 20 nucleotide protospacer will always find some identities somewhere else on the wheat genome as well. Okay? And this is indicated. This is 37, a lot of off-target sites, 28, 50, 55. So this is the reason it is recommended that design at least 8 to 10 protospacers and test them. Now, which one will you select for making the gRNA? Obviously, the one which has the minimum number of off-target sites. So your protospacer selection is this one. The second one would be the 17 one. Or design more protospacers and look for those which have a lower number of hits with the nucleotide sequence of the target genome, that is triticum, estimum, or wheat in this case. Do you understand that? Is it clear?
Yes, sir. Clear.
Okay. Okay. So now I will move ahead. Now you can actually download results file. Okay. So these are the blast results I already told you. So what you do is you click on this green area. Okay. Click on the green area and you go to the next tree and then download result file. Click on that and you will get the nucleotide sequence and the position. You scroll down actually this page and find out all the 11 positions where your protospacer is actually binding. You see, here, here, here. But look at that 11 positions. Your protospacer is 20 nucleotides long. It's binding at this position from nucleotide number 1 to 20. At this point, it is binding from nucleotide 1 to 18. So, but because it is 20 nucleotides, 19 and 20 may have some dissimilarity, but still 18 can bind. There's a 16, a lower efficiency of binding, a little higher efficiency of binding, a complete efficiency of binding. So you scroll down and you will find, you can see here, similarity from 1 to 20 with a mismatch at position here. Okay. So what I will do is that I will definitely go and select the first one. Okay. This has a minimum number of matches, just 11. So you can click on the second and it will show you where in the genome your protospacer is going to bind, non-specifically and specifically. You have a look at that and what is the length of that specificity. Is it important or not? The shorter is the specificity. It means you should worry less about that. Okay, so you should pick definitely the one which is having high degree of specificity for the target one. Okay. Now, once you have decided or selected the protospacer, wasn't it easy that you design the protospacer and you screen your protospacers by making a Word file, copying the sequence in the FASTA format and put into Ensembl plot and search against the target genome of that particular plant, and it will tell you which one is the best protospacer. And now you selected the protospacer which has the minimum number of non-specific binding sites. Was that simple or complex? Hello? Anybody? Press your space bar. It's quick to
Yes,
answer.
sir.
Was that simple or complicated? Selecting a protospacer.
So it was simple.
It was simple, yes. Things are really simple. Only there should be a chance that you learn. Okay? So if you learn, the things become really simple. So vector construction and transformation starts. Once you have selected the protospacer, the next thing is that you design a gRNA. Okay? So that is vector construction, designing of the gRNA. So what you do? You have a single vector or two vectors. Mostly people are using, for plants, people are using two vectors. One vector codes for the Cas9, the protein, okay? And you can have any constitutive promoter like polymosaic virus promoter here, CFD promoter, and you can put in one vector which has got a left border and a right border on it, so that your Cas9 protein gene can be integrated into the host genome. Okay, not a good approach really, but because this is how it was done initially, so I am elaborating it, that you should express Cas9 protein in the host system. You can express it stably by using a vector which has left and right borders. And you have gone through the transformation lecture and agrobacterium mediated transformation lecture. I gave it quite in detail. Okay. So if you put the gene cassette in cloning vector or in the agrobacterium plasmid vector, you can integrate this gene into the host genome and it will always express the Cas9. But do you think the expression of Cas9 all the time in a plant system throughout his life is a wise thing? No, it's not wise. It's better to go for transient expression. Okay? The protein is produced only for a short span of time. And when the editing is done, the protein is gone. So plants don't have any harsh effects of that. And the second vector that you have got is for making the gRNA. So you have the vector, second vector, which already has got a U6 promoter, a multiple cloning site here, and then gRNA scaffold. What is gRNA scaffold? It's a CRISPR RNA at this place and tracer RNA at this place. So it's a single component CRISPR RNA and tracer RNA. What the gRNA has got? It has got a protospacer, a CRISPR RNA, and a tracer RNA. That's all the DNA has got. So you have already in the vector the gRNA scaffold, that is tracer RNA and CRISPR RNA. So you have multiple cloning sites. You just clone your target here. What is your target? It's the 20 nucleotides long protospacer. So what you do? Your vector has got MCS. You select the clone restriction sites there. And you design a primer of 20 nucleotides long, representing the target sequence. But on that 20 nucleotide sequence, on the 5' end, you add one restriction site. On the 3' end, you add the second restriction site. Just cut the double-stranded DNA fragment of that synthesized DNA with the respective enzymes. Cut the vector with the same enzymes and clone that here. So you got a vector which will express the gRNA having that protospacer. Now, what people are doing actually, they are following the natural system. What they are doing, this is the gRNA scaffold, which is shown here. And this is the target, which is a single protospacer. So what they are doing is they are inserting more than one protospacer. And if you remember, when I showed you the first time, the cluster of phage sequences on the E. coli genome, they were separated by a repeat, a palindromic repeat. Okay, so what you do is you can put multiple number of protospaces separated by some palindromic repeats. Okay, so you will, if you have three repeats, three protospaces separated by palindromic repeats, so you will get three different types of gRNA. One gRNA which is having this protospacer, another gRNA which is having this protospacer, and another gRNA having this protospacer. Okay, so you are targeting multiple positions on the target sequence simultaneously. Okay, so you don't have to put a lot of different vectors for doing mutations at different points. Okay, if you remember, in this sequence where we started, we need to do cut at two positions. Okay, so what we can do is that we can actually design two protospacer sequences upstream to the gRNA scaffold. Just two sequences. We will get two gRNAs. Okay, if we are using CPF1, then we will be designing four gRNAs, two for a staggered cut before the mutation and two for a staggered cut after the mutation. So you can put four protospaces on the same vector and it will produce four different gRNA molecules. Is it clear or not?
Yes, sir, it's clear.
Okay, so this is how I have shown here actually the plasmids which are available. So what you can see is, you can see a left border here, a right border here. So you can see that Cas9 is here. So you transform this vector, which has got Cas9, directly by agrobacterium mediated transformation or by ballistic, whatever you like. You have a second vector, and on the second vector, you have chimeric sgRNA. What is a chimeric sgRNA? This one. This one. This is a chimera of different protospaces. Okay, so you may have a gRNA here having a single protospacer, or you may have a chimeric gRNA here, which can code for more than one, and you will get more are the number of protospaces on that sequence, more will be the gRNA produced. Okay. And what is the difference between gRNA and sgRNA? G stands for guide RNA. Guide RNA means the protospacer is actually a guide to take the whole protospacer, CRISPR RNA, and tracer RNA complex to a very specific site on the target sequence. So it is a guide RNA. If it is a single protospacer with the guide RNA, it is called sgRNA. Or you can say gRNA, it means the same thing. Okay, so use whatever word you want. But if somebody asks you what's the difference between sgRNA and gRNA, you will tell it is no difference. Okay? Now, Let's look at the strategy in plants right from the beginning. What did you do? You took the nucleotide sequence of the target. You designed 20 base pair protospacers close to the PAP site. Okay? And then you used online tools to minimize off-targeting. You analyzed the protospacer for the off-target sites and you selected the ones which give you minimum number of specific target sites, non-specific target sites. So this is one part that we did very well. Okay. Design sgRNA. You put your protospacer upstream to the sgRNA backbone. That is the gRNA scaffold. Okay, having CRISPR RNA and tracer RNA. You just clone that here. Okay, that is designing sgRNA. And then you trigger this U6 promoter. It will produce this RNA, which has the guide sequence, that is the protospacer, plus attached to it the CRISPR RNA, and attached to it with a loop, you remember, primer, GAAA loop, okay, and then the tracer RNA. This is now complete gRNA which will be produced. And then again the rule. First nucleotide guide sequence is a G if U6 promoter is used, or an A if U3 promoter is used. Okay. Guide sequence should match the target except for the first nucleotide, 5 prime G or A. It means the same thing. That does not have to match. Okay. Assemble Cas9 sgRNA construct. You can have Cas9 on one vector and sgRNA on another vector. That's a binary system. Or you can have the Cas9 or sgRNA on the same vector system. All your expression should be transient. Okay, I won't recommend you to go and do a stable integration of Cas9 into the host genome. It will be a disaster, really. Many off-target sites will be generated continuously over the lifespan of the plant. Okay, Cas9 should express for a short time. sgRNA should be expressed as a short time. There should be a genome editing event and then all gone. Genome is edited, nothing changes afterwards. Okay, so it depends in which lab you are, what are your objectives, and go for the single vector or binary vector system. Okay, it's not complicated. I'm telling you, this is so easy job. It's a very easy job working with CRISPR-Cas systems. Okay, just have to select the protospacer, clone it, and transform it. You can transform the protoplasm, you can transform through agrobacterium, or you can do it through microprojectile. Okay, particle bombardment. And then just you need to screen. And you do that by amplicon sequencing. You know the sequence of the target, so you can design the primer, and around the mutation site, you can decide the primers forward and the reverse. Do the sequencing with forward, do the sequencing with reverse, okay, and then you get the amplified product and sequence those amplified products. Do they have mutation, the desired mutation that's made, or not? That selection. So you select it. Or if you are very rich, you have very good projects, you can just go for NGS sequencing. Sequence the whole genome after DNA modification. So It will show you whether your target gene has been modified or not, and what are the other places where the non-specific mutations have occurred due to non-specific editing, genome editing. That is the off-target side. Okay, I hope the system is very much clear in case of plants. Now we are going for animals. Okay, gRNA designing for animals. Very simple. Go to Ensembl. In that case, we went to Ensembl plants. In this case, you just go to Ensembl, click on all species, and you will see the animals here. The human is there, mouse, zebrafish, and many other, cat, chicken, chimpanzee. So what do you want to work with? This is your decision, okay? So just like in plants, you select the source here, and then you just type the name of the gene that you want to modify. TRP53, one of the genes involved in cancer. You want to back mutate it. You want to remove the SNPs from there. Okay? First, you need to retrieve the nucleotide sequence of that TRP53. And when you search that in that genome, after selecting the genome, you write here the name of the gene and you come up with all the genes. These are the variants having the SNP. This is the, on the top is the actual gene. So you click on the actual gene and you come up with a page which shows show transcript table. What does it mean? You know that eukaryotic genes, they have introns and exons. So you want to see the exons from where the transcripts are produced. Okay, click on show transcript table. Okay, so it will tell you these are the transcripts. So you can just click on TRP53, the first one. Okay, just click the one, and what you get, this is just, it gets highlighted first, and then it will show you all the exon nucleotide sequences. Now figure out, identify which exon has the mutation that you want to repair. Just pick that exon. For example, this one. You just click here and copy this sequence from there. Copy and paste on Word file. This becomes your target sequence. Okay? In case of plants, you can just pick the genes. Okay? In case of animals, maybe you have to look for the genes which have mutations. So you just give the name of the gene and pick the transcript variants and select the exons which code for those and select the particular exon where you want to do the mutation. Okay? And then you don't do yourself the designing of the protospaces. You can do it actually, but don't do it. In case of animals, the things have been advanced at a much more higher level than in case of plants. So the softwares that are available, like CRISPR design, is one of the software. You write it on the Google search, CRISPR design, and you can open up this page. Search name, you can name the search. You can give your email address, okay? And then tell in which animal you want to make this protospacer sequences. Design the protospacer sequences. Okay, after putting the name of the gene, your email address, you can paste here the sequence of the exon which has got the mutation. And this is the same exon that we picked from the TRP53 search. Okay, paste the sequence. Tell that you want to prepare the protospaces in this region, okay? And then agree and submit. So what you get, you come up with screen. And if on the top you look at this bar, it is processing your work. This is about 1/10 of process that has been completed. It has to go through all this length, okay? And after all this process has taken place, you can click here, guide and off targets. So when you click here, you will get this thing. All these are 20 nucleotide guide protospaces that the software has developed on that particular target sequence. And then you will see the score values also there, that how good is this, okay? Now, it is 97, means very good. 94, a little bad. 91, very bad. 89, worst. 73, no, not at all. So obviously, on the top, you will see the best protospacer sequence selected by the software. And at the same time, on this CRISPR design, for the first one, this first one is selected. So you click on the first one and you come up with quality score 97 on the top written here, then the guide sequence written here, okay? On target locus, chromosome 17 at this position, which is a TRP 53, okay? And 10 important information. Number of off-target sites, 25. Okay, so it means that if you take this protospacer and use this for making a GR for the human genome, there are 25 sites on the human genome which will be modified using this protospacer on the guide RNA. And then what you do? You select the second one and click on that. When you do that, the second one has a score of 94. And if you come down, number of off-target sites, 40, much more than 25. And at the top, it is also showing you the whole genome region where these protospaces are actually developed. What is this part? This is actually TRP 53 and the exon that you selected. All those protospaces, they are designed on that exon sequence. You see a lot of them. So when you hit the second one, it will be shown up there as blue colored. Okay, so you can see this is the protospacer position and it has got 40 of target sites. And this is how you select the best one. Okay, just like you selected in the plants, there you got one of the protospaces for 11 of target sites. Here, the minimum one for the first one is 25. You have to go clicking one by one each of the protospacer sites and look here how many are the off-target sites and select the one which is having the minimum number of that. Okay, now you come back to the analysis page. You have done with this. Now you design the CRISPR-Cas. CRISPR-Cas whole system does not require a single gRNA. It always requires 2 gRNA. One after the point of mutation and one before the point of mutation. So this is the grid or box. If you select here now, after selecting the protospaces, okay, so you will get, you can know from here that which one was selected by you. Okay, so you just click on there and case analysis, and these are the same protospaces which appear on the previous. If you select this one, it will suggest to you, okay, this one is the one that you selected, the most appropriate second protospacer for cutting at the second site to release this part of the DNA fragment is this one. So this is the pair of gRNA that you should design. Okay. Now, if you click on any one of these two, you come up with the nucleotide sequence and their position. This is for one protospacer, this one, and this is for the second protospacer. Okay, so your cut will be here, this fragment will be removed, and you need to insert an identical fragment along with a mutation to replace that in the target gene. Is it clear? Simple. It's much simpler in case of animals. The softwares are much more developed and accurate. Okay, so you need to learn. You need to do some practice, and this is shown in slides that how you should go for that. Okay, and finally, what you do? You make the gRNA construct and the Cas9. You can take the embryonic stem cells, okay, and put those embryonic stem cells after transformation. Select the clones which are having your mutated genes, I mean edited genes. You can transfer those embryonic cells into the surrogate mothers and look at the progeny which has got that genome edited subject. Okay, so this is the strategy. Now, what has been produced? What are the disadvantages of CRISPR-Cas system? I have been mentioning again and again. Number one, you need a reference genome. The genome should be there in the Ensembl. Okay, if there is no genome available, you cannot determine the off-target sites. Then off-target sites, you always see they should be, if it should be 1, they are 11 or 25 or 40. So off-target sites are the biggest problem. And there are some other ones which are written here, so you can just read them. These are the drawbacks of genome editing. Okay, and now this is the first crop that was modified. The long shelf life tomato was already produced through RNAi approach. That's an old work. But when the CRISPR-Cas technology came up, they did use the same technology to modify not only pectate lyase, which is responsible for ripening of the cell wall of tomato. They also studied polyglucoronase 2A and beta-glucanase. And they identified that pectate lyase is actually the enzyme which is involved in late ripening. If you disrupt this gene, the tomato has late ripening capabilities, while the others, they actually change the color of tomato or size of or weight of the tomato. So these genes, they are not important in late ripening, but pectate lyase was the enzyme. So this is the publication in Plant Physiology, which utilized the CRISPR-Cas9 technology to modify this trait. Okay. Then some of the scientists, they use the technology for good things. Some of them, they start doing the things which are banned. So this was an example where Jiankui, he actually targeted some mutations in the human embryos, okay, which was illegal, okay, and you might have heard about that in the newspaper. It was done in December 2018. In a conference, he presented the work openly at open forum. So people do bad work, people do the right work too. But this was a very bad example of working on the humans. CRISPR-Cas technology is not yet permitted for humans. Okay, remember that. Now, the thing that we addressed in the very first point, if we are generating GMOs, then we undergo several regulations. Okay, so this is the most important fight in the court. Okay, genome editing is GM. They started saying that because people started replacing or putting the whole gene cassettes into the organisms, animals or plants. So that became a GM. So everybody started thinking that way. But DNA repair, that where you are mutating back the SNPs, it's a repair. You're not inserting it. So nobody cares about that. And the case was in the court, and European court has decided that genome editing is equivalent to GM. So it has to go through all the regulations, which is the biggest drawback for everybody. Okay. So if you talk about these GM technology or the genome editing technology with males and females, here is the statistical analysis. The females are against these GM technologies and genome editing technologies much more than the males. Okay, nobody knows why, but males a bit favor that technologies of GM crops and genome editing, but the females at all these points, you can see the females, they have voted against that rather than the support. So with this, I'm finishing my CRISPR-Cas9 lecture.